Good afternoon. I hope you guys aren't feeling the postprandial blood sugar drop that I do, but we'll have some good energy here. So Arbor is a clinical stage next generation gene editing company. We're co-founded by Feng Zhang, David Walt about 8 years ago. Since then, we have built a robust set of gene editing technologies that allow us to really pick targets and then decide, screen against a variety of gene editing approaches to pick the right approach for the right target. Our first wave of programs are in the clinic today, and that's in primary hyperoxaluria, which is a rare liver pediatric disease. Our second wave of programs are really focused in the CNS, where we feel there's a real opportunity to move gene editing out of the liver and into the CNS, where we can really take advantage of all the great biology that's been done over the past decades and point us to some really interesting targets in the CNS. Our first wave of programs are targeting ALS. We have partnered multiple programs, particularly in the ex vivo space, where we are allowing folks, they take our technology to do whatever fancy gene editing they need to do to their cells and pushing those forward. So Allogene's an example who's using our technology in their autoimmune program. We are a very capital efficient company and have tried to make sure that everything we do, we are focused on small number of things in our pipeline and focused on bringing those as quickly as possible into the clinic. I will skip this one because we are all cell and gene therapy advocates at this meeting. So yesterday we actually announced a collaboration with Chiesi Pharmaceuticals. This is they're about a $4 billion in revenue Italian company, privately owned. And our partnership with them is two components. First is a license agreement on our lead program, which is in the clinic, ABO-101. Secondly is a multi-target research collaboration. And you can see some of the economics. We're very excited about this, to partner with a company that has a focused rare disease group that is generating hundreds of millions of dollars in revenue with that group and really work very carefully and with patients to drive these novel therapies to the rare disease population. So we're very excited to have that partnership. We do have a very experienced team. This is important for biotech, particularly in gene editing, where we need to focus and we need to get things through to the clinic as soon as possible. I mentioned this a little bit. So the way we have done our work over the last 8 years in really building this range of editing approaches is really sourced from nature, but then importantly, you can find lots of editing things in nature. There's millions and millions of different approaches. However, turning those into editors that are relevant and work in human cells is not trivial. In addition, you need to find ones that have particular properties that are helpful for the type of editing you're trying to do. And so it does allow us to be, we're very good at this now with our team. They can very quickly identify these novel approaches and bring those via different engineering approaches to very therapeutically relevant levels of editing. So we have 4 types of editing we like to do. We feel this covers the broad gamut of things you want to do within genetic diseases or even beyond that in all diseases. So you can knock down proteins where you have too much of something. You can do what we call RT editing, where you can couple a reverse transcriptase to a nickase to rewrite sections of DNA, and I'll tell you a little bit about that. Excisions, where you can remove any sort of repeat region, like some of these hexanucleotide repeats in the CNS. And finally, insertions, where you can utilize the technology to insert larger chunks of rewrite pieces of DNA. Importantly for us has been making sure we have very small technology. So our technology can fit into any delivery modality, which allows us in particular to move outside the liver and utilize approaches other than LNPs for delivery. You can see our pipeline here today. 4 programs in the CNS, 3 tackling ALS and undisclosed using a compact RT editor. So this is an RT editor that can fit into a single AAV. And we've got an interesting target in the CNS there. I'll the interest of time, I'll skip our lead program that's partnered, but we have, we're very excited about this one. We've dosed several patients and are now working closely with Chiesi on this one. Delivery is an important piece of the puzzle. So in the CNS today, there is no non-viral approaches for delivery that have been shown to work in NHPs that we've seen. If someone sees it differently, we'd love to hear about it. Which means that we need to utilize AAV. And there are a variety of approaches with AAV one can utilize. You've got sort of the traditional AAVs that you can use via local delivery, as many companies are doing, or you can utilize novel capsids. So we do have a partnership we've announced with 4DMT to utilize their novel capsids, and we have worked with others as well. So there's multiple approaches for delivery, but as a company, we are delivery agnostic. We don't really care what delivery approach we use as long as it's one that is safe and gets us to where we need to get to. And that way we don't spend precious capital building our own delivery portfolio, our delivery platform. CNS. A lot of patients. I mean, it's pretty obvious there's a lot of patients in CNS, but for us, we wanted to start somewhere we could actually be a little more tangible on value. So the ALS is a great starting spot. We know we can target spinal motor neurons today utilizing AAV, and we also know that there are a mix of genetic and sporadic cases of ALS, both of which have very interesting targets for creating therapeutics. From there, we can begin to move more broadly into other diseases. ALS particularly, you've got the two biggest genetic drivers are C9orf, which is a hexanucleotide repeat version that causes the disease, and then SOD1. And then the genetics are only about 10% today of the ALS population. The majority of patients are actually sporadic, unfortunately. And as anyone who has known someone with ALS, it is horrible. Once you've got it, it is, you're just kind of hoping for the best. There's no treatments, and you hope you have a slowly progressing version because it's deadly. So one of the approaches we're taking is looking at SOD. This is the second most prevalent genetic version. And so what we have to do is we need to see, okay, do we see significant DNA editing that's highly potent? Indeed we do. Does that result in the right biology, which means your SOD protein goes down? Indeed it does in a dose-dependent manner. And then finally, if you move to in vivo, do you see that same recapitulation of the data that you saw in vitro? And indeed we do. And this has been one of the things I think that we find what's great about gene editing is if you can get it to the cell in vivo, you recapitulate what you see in vitro. And unlike I worked in small molecules for a long time, that is never the case in small molecules. So it's lovely to have something that is completely predictable and translatable. The other piece that's important, there's a humanized SOD mouse model where you can really begin to look at survival. So in this case, we ran the experiment with the mice, and we saw survival well over 100 days. Now, there is a currently approved ASO, tofersen. The survival that they saw in the same model was 23 days. So significant impact on survival. Why? Well, I think could be many reasons for it. One particular reason is you think about ASOs, within a particular cell, you're going to knock down some portion of the toxic protein, but not 100%. With a gene edit, it's binary. You either knock it out completely or you don't. And so that may be one of the reasons we do see pretty amazing efficacy in this particular model. Now, this is important because our approach is to really take multiple targets to ALS so that we can utilize the same delivery, begin to think about some of the regulatory benefits of approaching it via platform, et cetera. Our second program I'll tell you about Stathman. So Stathman is a microtubule binding protein. It controls axonal outgrowth, and it's one of the most abundant proteins in neurons. It is regulated by TDP-43, and it is what happens in patients. So this is for all sporadic patients. They get these TDP-43 aggregates. TDP-43 is critical for splicing, and when you have these aggregates, it does not cover these cryptic splice sites, so you get missplicing and truncated proteins, which are dysfunctional. When we look in patient cells, so if they have in the top column there, see the cells that are very happy neurons, that have beautiful processes. When they have the TDP43 pathology, the processes are gone. We can restore those as we gene edit out the cryptic splice site. So we go in and remove that cryptic splice site. And this is something that our technology is uniquely suited to that we're using here, because with this particular editor, actually, when it cuts, removes about 20 nucleotides as it cuts. So it's really nice for removing larger pieces of DNA. Now the question is, how does that translate to in vivo? There is a Stathman humanized mouse model we can look at. We see really nice dose dependency on editing. This is looking at editing the whole brain. Importantly, though, what does it mean on the truncated? Do we see reduction of the truncated message? Indeed, we do. In that second one, you can see it's greatly reduced in a dose-dependent manner. And importantly, we see increases in that full-length mRNA, which results in increases in the full-length protein. So it's very exciting because, again, it's recapitulating biology that we see in vitro in patient cells that's being recapitulated in the animal. And next steps to hopefully recapitulate this in the human. We are very excited to continue to push these programs forward, and we are targeting moving into the clinic with these programs over the next 18 months or so. So just a little bit on compact RT editor. So this is as I mentioned, a single editor that fits nicely within an AAV. I'll skip the overall biology of an RT editor, but allows you to rewrite DNA. So in the case of traditional editing with Cas9 coupled to a Maloney reverse transcriptase, it's big. It's much larger than what you can fit into an AAV. In our case, we're able to identify both a nickase and a reverse transcriptase that are both very, very small, very, very novel, and are about easily fit within a AAV, which means you can deliver a single AAV for RT editing, which is fantastic. No one has demonstrated this in vivo, and we're pretty excited we have. So we were able to see high levels of editing, not only in the liver, but also in the heart and in the brain with this compact RT editor. So in post-mitotic cells with a single AAV. So this opens up a tremendous number of interesting targets in the brain or other areas where you can only use AAV for targeting today. So we're very excited about this. The other piece that's quite interesting as you deliver via AAV is the editor's there, right, for a long period of time. And you do begin to see that over time, you see that editing increase, and then it plateaus. And we've been able to monitor off targets and other pieces over time and see some real interesting and good safety there as well. On the large insertion side, so we have an approach called synthposons, which again utilizes nickase and reverse transcriptases to be able to write larger chunks of DNA in an all RNA-based approach. You can see here some of that data. We've been able to even see measurable rewriting up to 800 base pairs in cells. And you can see that falls off as you extend, which is not surprising because you're dealing with a lot of different components in the cell that have to work well. Your RT has to remain on there for a long period of time. Think of it as kind of PCR within a cell. And it's very exciting to be able to see this level over 800 base pairs. This is using an RNA-based template approach. So we continue to move forward with our liver pipeline with that partnership with Casey I mentioned, and then in the CNS moving ahead very quickly to generate pushing towards DC nomination towards the end of this year, and then IND filings as soon as possible thereafter. And in summary, our goal is to have multiple submissions over the next 3 years, and that's enabled by not only our technology, but also our amazing team we have. So thank you very much.